Welcome. In this session, we'll be discussing about the retinal pigment epithelium and the Bruch's membrane. In the previous sessions, we have discussed about the vitreous and the neurosensory retina, and in the following session, we'll discuss about the choroid as well as the retinal circulation. The retinal pigment epithelium is a monolayer of pigmented hexagonal cells derived from the outer layer of the optic cup. It is continuous with the pigmented epithelium of the ciliary body at the ora serrata. There are approximately 5 million retinal pigment epithelial cells, and after birth, the retinal pigment epithelial cells are post-mitotic, that is, they do not divide following birth, and number of RPE cells thus remain stable throughout life in the absence of disease. And the number of photoreceptors interacting with each RPE cell is said to be around 30 to 45. The number of RPE cells per unit area is highest in the fovea and decreases in the periphery, and so the RPE cells in the fovea are taller and thinner than those in the periphery. The RPE cells, like most other epithelial cells, are said to be polarized. The cells have an apical region towards the neurosensory retina and a basal region towards the choroid. The apical region has villi, which surround the outer segments of the photoreceptors, with whom they are intricately linked metabolically but without any direct physical connection. The lateral surfaces of adjacent cells have three types of junctions. The zonuli occludens, or the tight junctions, are located on the lateral membranes near the apical region and is present all around each cell, forming the outer blood-retina barrier. And this is very important to remember. The zonula occludens thus prevents transepithelial transport across the RPE cell through the paracellular pathway. Zonuli adherentes, which are located in the lateral plasma membrane basal to the tight junctions, which have attachments to intracellular actin filaments and are involved in maintaining cellular shape. Gap junctions between the lateral cell membranes of adjacent RPE cells play a role in transferring molecules and electrical potential between the cells. The basal region of the RPE cell has infoldings of the plasma membrane, which is not followed by the basement membrane. These infoldings increase the surface area of the basal plasma membrane for transfer of molecules. The retinal pigment epithelium is attached to the Bruch's membrane, of which the retinal pigment epithelium basement membrane is a part. Bifocal adhesions between the basal plasma membrane of the retinal pigment epithelium and the retinal pigment epithelium basement membrane. The nucleus and mitochondria are located in the basal region of the RPE cell. Apart from the routine set of intracellular organelles, RPE cells are particularly rich in melanosomes, phagosomes and phagolysosomes, and microperoxisomes. Melanosomes are spheroidal cytoplasmic organelles containing melanin, which absorbs light, and melanin concentration has been found to be higher in retinal periphery than in the center. Melanosomes are mostly present in the cytoplasm of the apical and central regions of the RPE cell. The phagosomes and phagolysosomes, which are involved in phagocytosis of disc membranes of the outer segments of the photoreceptors, and we'll be discussing this process further. And microperoxisomes, which have antioxidant functions and are involved in detoxifying free radicals and lipid peroxides. The RPE cells have a number of functions which we will be discussing individually. As we have mentioned, melanin in RPE cell absorbs light passing posterior to the photoreceptors, thereby minimizing reflection of light back into the neurosensory retina, thus reducing scattering of light in the neurosensory retina, which may potentially interfere with visual acuity, and also reducing oxidative damage by light in the neurosensory retina. And as we have also mentioned previously, the tight junctions between the apical regions of the RPE cell forms the outer blood-retina barrier, preventing free diffusion of molecules between choroid and retina through the paracellular pathway. Another important function of the RPE is maintaining a fluid-free subretinal space by removing water from the subretinal space, and this is said to be primarily responsible for retinal adhesion. There are other factors which also contribute to retinal adhesion, and we will briefly discuss them here. The interphotoreceptor matrix binds to both the neurosensory retina and the retinal pigment epithelium and has an adhesive nature which prevents physical separation of the neurosensory retina from the retinal pigment epithelium. The interdigitation between the outer segments of the photoreceptors and the microvilli of the RPE cell also contributes to retinal adhesion. And fluid pressure, both the hydrostatic force of intraocular pressure and osmotic force of choroid, contributes to retinal adhesion. Following retinal detachment repair, it may take several weeks to restore the interphotoreceptor matrix and interdigitations between the outer segments and the microvilli. The retinal pigment epithelial cell is responsible for transfer of nutrients and ions to and removal of waste products from the outer retina, and this occurs through transcellular pathway but not through paracellular pathway because of the presence of the tight junctions. Sodium potassium ATPase is located in the apical membranes of the RPE cells, unlike other epithelia where it is located in the basolateral membranes. And this sodium potassium ATPase actively transports sodium towards the retina and potassium towards the choroid. The glucose transporter GLUT1 is located in the apical and basal cell membranes of the RPE cell, transferring glucose from the choroid to the retina, and the waste product lactic acid is eliminated from the subretinal space by monocarboxylate transporters. Water moves along its net osmotic gradient, moving from the subretinal space towards the choroid through aquaporine and chloride channels. And as we have mentioned, this movement of water from the subretinal space towards the choroid is primarily responsible for retinal adhesion. The RPE cells also phagocytose the outer segments of the photoreceptors, including their membranous discs. The rod shed disc at light onset and is most vigorous at dawn, and the cone shed disc at light offset and is most vigorous at dusk. And it has been estimated that each photoreceptor shed approximately 100 discs per day, and so each RPE cell which is in contact with approximately 40 photoreceptors phagocytose approximately 4000 discs per day, or approximately 200 million discs in an 80-year-old lifespan. Since each photoreceptor contains approximately 1000 discs and shed approximately 100 discs per day, the outer segments of the photoreceptors are renewed every 2 weeks. Recognition of the outer segment of a photoreceptor by retinal pigment epithelium is a receptor-mediated process. And following this recognition, pseudopods in an RPE cell surround and engulf the tip of the outer segment in a phagosome. The phagosome moves to the basal aspect of the cell, and then lysosomes fuse with the phagosome to form phagolysosome. Lysosomal enzymes, particularly cathepsin D and cathepsin S, degrade the contents of the phagolysosome, and lipids in the phagocytosed discs are recycled to the photoreceptors for making new membranous discs. The remainder gives rise to liposuction granules, which are undegraded residual bodies resulting from the phagocytic activity within the phagolysosome. Liposuction granules may accumulate in the cytoplasm of the RPE cell or may be excreted by the RPE cell and accumulate between the basement membrane of the RPE cell and the inner collagenous zone of Bruch's membrane, which we will discuss as drusen. Liposuction generates reactive oxygen species, inhibits phagocytosis of disc membranes, and promotes cell damage. Liposuction granules are also responsible for the signal obtained during the diagnostic procedure called fundus autofluorescence. Increased fundus autofluorescence indicates preferential loss of rods and thus correlates with delayed dark adaptation and decreased scotopic sensitivity. The RP cell also participates in regeneration of the chromophore 11 cis retinal, which is called the visual cycle. The visual cycle takes place partly in the outer segment of the photoreceptors and partly in the retinal pigment epithelial cells and is similar for both rods and cones. Upon capture of a photon by rhodopsin, 11-cis retinal is converted into all-trans retinal, and this is called photoisomerization. All-trans retinal causes conformational change in rhodopsin to metarhodopsin to initiate the phototransduction cascade. All-trans retinal then detaches from the metarhodopsin to allow another cis retinal to attach to rhodopsin. An 11-cis retinal attaches to an intramembranous portion of rhodopsin. All-trans retinal detaches from metarhodopsin and is transported to the cytoplasm by ABCA4 and ATP binding cassette transporter protein. In the cytosol, all-trans retinal is converted to all-trans retinol by retinal dehydrogenase. All-trans retinol then escapes from the outer segment to bind to interphotoreceptor retinoid binding protein in the interphotoreceptor matrix. The interphotoreceptor retinoid binding protein or IRBP in the interphotoreceptor matrix and the cellular retinal binding protein 1 or CRALBP1 in the RPE cell bind to the retinal intermediates in the visual cycle so as to prevent damage to the photoreceptors and the RPE cells from these highly reactive retinal intermediates in the visual cycle. IRBP then transfers all-trans retinol to CRALBP1 of RPE cell, and all-trans retinol remains bound to CRALBP1 while being converted to 11-cis retinal in the RPE cell. So retinoid intermediaries remain attached to CRALBP1 in the RPE cell while being converted to 11-cis retinal. Apart from the visual cycle, the RPE cell also obtains retinoids from two other sources, from blood and from phagocytosis of outer membrane discs of photoreceptors. In the RPE cell, all-trans retinol is first esterified by lecithin retinal acyl transferase to all-trans retinal ester. All-trans retinal ester is first hydrolyzed and then isomerized to 11-cis retinol by the enzyme RPE65, also known as retinoid isomerohydrolase. And then 11-cis retinol is oxidized to 11-cis retinal by retinal dehydrogenase. 11-cis retinal is then released by the CRALBP1 and escapes from the RPE cell into the interphotoreceptor matrix where it binds to IRBP. And IRBP then transfers 11-cis retinal to the outer segment of the photoreceptors to join with a regenerated aporhodopsin molecule, and this regeneration of aporhodopsin molecule, as we have discussed in the previous session, is mediated by rhodopsin kinase, leucovorin and arrestin. The RPE cell is also responsible for formation and maintenance of the Bruch's membrane and the interphotoreceptor matrix. Interphotoreceptor matrix is also contributed by photoreceptor inner segments. The RPE also secretes growth factors, and two of the important ones are pigment epithelium derived factor, which is released from apical surface of the retinal pigment epithelium and provides a neuroprotective and anti-angiogenic environment for the photoreceptors. Vascular endothelial growth factor is released from the basal surface of the retinal pigment epithelium and maintains choroidal vascular endothelium. The retinal pigment epithelium maintains a relatively immune privileged site in the subretinal space by secreting factors such as TGF-beta, but it can initiate an innate immune response to pathogens and damaged molecules by expressing toll-like receptors and complement factors. Biochemistry and metabolism of RPE cells is intricately linked to their functions, but we'll discuss them separately. Retinal pigment epithelial cells utilize all methods of glucose metabolism to generate energy, such as glycolytic cycle, Krebs cycle, and hexose monophosphate shunt. Glucose is the main source of energy, and glucose is not converted to glycogen for storage. Lipids constitute 3% of weight of the RPE cells, and 50% of lipids are phospholipids. One source of phospholipids are the phagocytosed disc membranes of the photoreceptors, from which the fatty acid docosahexaenoic acid is recycled back to the photoreceptors. Levels of saturated fatty acids are higher in retinal pigment epithelium cells than in the outer segments of photoreceptors, and these saturated fatty acids are involved in retinal metabolism as well as energy generation. Melanosomes of the RPE cell contain melanin, and RPE is the earliest site of melanogenesis in the body. Melanin concentration is higher in the retinal periphery than in the center, and this contrasts with the density of the RPE cells, which is higher in the center than in the periphery. With age, fusion of melanosomes with lysosomes may lead to loss of melanin pigment, leading to loss of fundus pigmentation, particularly in the retinal periphery. In addition to absorbing light, as we have discussed, melanin also has other functions such as promoting proper neuronal migration in the visual pathway during development, which is exemplified by albinism, in which there is a congenital absence of melanin synthesis, and albinism is associated with foveal aplasia and with the majority of optic nerve fibers crossing in the optic chiasm. Melanin also stabilizes free radicals and bind drugs and toxins such as hydroxychloroquine and thioridazine, which are known to be retinotoxic. Melanin is also involved in converting light energy to heat energy during laser photocoagulation. Choroid is spared from burns during laser photocoagulation, as choroid has a very rapid blood flow which acts as an effective heat sink. Antioxidant mechanisms are present in the retinal pigment epithelium to control oxidative damage. RP is prone to oxidative damage as it is present in a highly oxidative environment because of the high oxygen concentration at that location, exposure to light, presence of abundant mitochondria, and a high concentration of polyunsaturated fatty acids in disc membranes of phagocytosed outer segments in phagolysosomes. Antioxidant systems are present both in the cytoplasm and mitochondria of the RPE cell to minimize oxidative damage. But it should be noted that carotenoids lutein and zeaxanthin are not present in the retinal pigment epithelium. Autophagy occurs in the RPE cell, and autophagy is a process of degrading the cell's own damaged intracellular components and recycling the degradation products. The autophagy products in retinal pigment epithelium are recycled to the retina and the choroid, and autophagy is important for maintaining intracellular homeostasis in a non-dividing cell. Phagosomes and lysosomes mediate autophagy, and dysregulation of autophagy plays a role in ARMD, photoreceptor degeneration in retinal detachment, and chloroquine toxicity. There is a standing trans-RP electrical potential resulting from asymmetrical movement of ions across the apical and basal membranes of the retinal pigment epithelium, and this is the potential which is recorded during electrooculography. On exposure to light, the EOG does not respond immediately, but 10 minutes later there is a depolarization on the EOG, and this is called the light response of RPE. And a chloride channel in the basal membrane of the RPE called bestrophin, which regulates an L-type calcium channel, is said to mediate this light response. Bestrophin is mutated in Best disease, and so EOG is abnormal in Best disease, but we must remember that EOG is not severely affected even in most other RPE diseases. Genetic defects of various retinal dystrophy have been found to encode proteins functional in the retinal pigment epithelium, and these include RPE65 in Leber congenital amaurosis, CRALBP in retinitis punctata albicans, and 11-cis retinol dehydrogenase in fundus albipunctatus. So in these entities, the enzymes in the visual cycle or the retinoid cycle are defective. In ARMD, degeneration or loss of retinal pigment epithelium causes atrophy of the adjacent neurosensory retina and choriocapillaries. And in retinal detachment, RP cells detach from the choroid and migrate from the subretinal space to the vitreous cavity through the retinal hole. and exposure of these RPE cells to vitreous leads to transformation of these RPE cells into myofibroblasts, and these myofibroblasts then proliferate on the surface of the retina, resulting in proliferative vitreoretinopathy, which is the most common cause of recurrent retinal detachment following retinal detachment surgery. Bruch's membrane is an extracellular matrix separating the retinal pigment epithelium from the choriocapillaries, and it consists of 5 layers with a total thickness of 2 to 4 microns. Starting from the RPE cell aspect towards the choroid, the 5 layers are the basement membrane of the retinal pigment epithelial cell, the inner collagenous zone, which is the thickest among the 5 layers of the Bruch's membrane, the middle elastic zone, the outer collagenous zone, which has intercapillary pillars as outward extensions between the choriocapillary lumens, and basement membrane of the endothelium of the choriocapillaries. This basement membrane is discontinuous with respect to the Bruch's membrane, since it is not present at the site of the intercapillary pillars, but is continuous with respect to the choriocapillaries surrounding the entire endothelium of the choriocapillary lumen. So the structure of Bruch's membrane is like a sandwich with a layer of elastic tissue surrounded by 2 layers of collagenous tissue lined by 2 layers of basement membrane. Functions of the Bruch's membrane include providing an attachment site for the RPE, as we have mentioned previously in this session, and this is mediated by interaction between integrin in basal plasma membrane of the retinal pigment epithelium and laminin in the basement membrane of the retinal pigment epithelium. The Bruch's membrane also allows passage of molecules between the retinal pigment epithelium and the choroid. Water flows across the Bruch's membrane along hydrostatic and osmotic gradients. The resistivity of Bruch's membrane to fluid or water flow increases with age due to lipid accumulation, which we will be discussing further. Solutes cross the Bruch's membrane by diffusion depending on their individual concentration gradients across the Bruch's membrane, and with aging, diffusion coefficient of the Bruch's membrane for these solutes increase, particularly for larger molecules such as lipoproteins, thus compromising nourishment of the outer retina and the retinal pigment epithelium. The Bruch's membrane stretches with increase in intraocular pressure and choroidal volume, but returns to its original state when these factors normalize. Endostatin, a fragment of collagen 18 present in the RPE basement membrane, provides an anti-angiogenic environment in the subretinal space. The RPE basement membrane is formed by the retinal pigment epithelial cell, and the choriocapillary basement membrane is formed by the choriocapillary endothelium. The inner collagenous zone, the middle elastic zone, and the outer collagenous zone are formed by both the RPE cell and the choriocapillary endothelium. Age-related changes in the Bruch's membrane include accumulation of lipids, and cholesterol-rich lipoproteins accumulate, which are particularly rich in apoproteins B and E. The lipoproteins with these apoproteins are sourced from plasma, where they act as carriers of various nutrients such as vitamins A and E, lutein and zeaxanthin for the neurosensory retina. These lipids are initially deposited in the middle elastic zone, then the inner collagenous zone, and then between the RPE basement membrane and the inner collagenous zone. And this pattern of deposition suggests that the lipoproteins, though sourced from plasma, are deposited in the Bruch's membrane from the RPE. And we should note here that the lipids in the membranous discs of the outer segments of the photoreceptors, which are recycled by the RPE, are not said to be deposited in the Bruch's membrane. And these lipids are the major contributor to age-related increase in thickness of Bruch's membrane. Calcification of the Bruch's membrane also occurs with age by deposition of calcium in the middle elastic zone, increasing brittleness of the Bruch's membrane. Bruch's membrane also accumulates oxidized metabolites such as age-related lipoperoxidation end products, advanced glycation end products, and complement components. And with age, there is a reduction in the proteoglycans present along with the collagen fibers in the Bruch's membrane. In age-related macular degeneration, the deposits which are typically found are the drusen and the basal linear deposit, between the inner collagenous zone and the basement membrane of the retinal pigment epithelium. The basal laminar deposit, which appears as a thickening of the basement membrane of the retinal pigment epithelium, and subretinal drusenoid deposits, which occur between the retinal pigment epithelium and the outer retina. And defects in the Bruch's membrane leads to development of choroidal or subretinal neovascular membrane between the choroid and the retinal pigment epithelium, and this is prone to occur in age-related macular degeneration, myopia, pseudoxanthoma elasticum, trauma, and inflammation. To recap the salient points, retinal pigment epithelium is a monolayer of pigmented hexagonal cells which are post-mitotic after birth and forms the outer limit of the retina. Plasma membrane of each RPE cell has an apical portion, a lateral portion, and a basal portion. The apical portion of the plasma membrane has microvilli which surround outer segments, and each RPE cell interact with 40 photoreceptors. The basal portion of the plasma membrane of the RPE cell has infoldings to increase the surface area for transport. Lateral portions of the plasma membrane of the RPE cell have tight junctions with adjacent cells near the apical region of the cells, and the tight junctions between the adjacent cells are present all around each cell. And these tight junctions prevent paracellular transport across the retinal pigment epithelium, thus forming the outer blood retinal barrier. RPE cells have melanosomes containing melanin pigment in the apical region of the cytoplasm. The basal portion of the cytoplasm contains the nucleus and the mitochondria, and other organelles include phagosomes and microperoxisomes. The functions of RPE cell include absorbing stray light with melanin, forming outer blood retinal barrier, transport of nutrients and waste products to and from the outer retina, phagocytosing outer segments of photoreceptors and their discs, regenerating the chromophore 11 cis retinal, maintaining the interphotoreceptor matrix and the inner portion of the Bruch's membrane, facilitating adhesion of neurosensory retina to the retinal pigment epithelium, and secretion of growth factors. The Bruch membrane is an extracellular matrix between the retinal pigment epithelium and the choriocapillaries. The layers of the Bruch's membrane from the RPE aspect to the choroidal aspect are basement membrane of the retinal pigment epithelium, inner collagenous zone, middle elastic zone, outer collagenous zone, and basement membrane of choriocapillaries. Functions of Bruch's membrane include attachment of retinal pigment epithelium to the choroid and transport of nutrients and waste materials to and from the RPE and the outer retina. Thank you for listening.